Okay, and speaking of genetic therapy, our next two grants have to do with genetic therapy for PWS. So there are two proposals or two projects that we're going to be supporting in this round that get at this question. You'll remember that everyone with PWS has the PWS region gene sitting there, but silent. So in deletion, you have one maternal chromosome that the genes are there but silent. In UPD or imprinting, we have two maternal chromosomes, the genes are there but silent. So as we've discussed, we've supported some initial work looking at whether you can turn those genes on. And the answer is yes, you can turn those genes on, but we still have a lot of work to do. So Dr. Jang used a drug that turned those on, and those studies are being followed up with the support of Levo Therapeutics to advance those drugs. We've also supported Dr. Lalande and Dr. Stormy Chamberlain looking at a different approach to turn those genes on, and they were able to correct or activate gene expression in neurons in a dish. But there are still a lot of really important questions that have to be answered before we can start thinking about doing this in people with PWS. So we have to understand whether activating these genes reverses the symptoms, which genes have to be turned on. So there were a bunch of genes in our PWS region. Do they all have to be turned on, or can just some of them be turned on? How much do they have to be turned on? When do they have to be turned on? And what might be, the consequences that, of turning those genes on kind of late in the process. So those are all really important basic feasibility questions we have to address before we advance genetic therapy for PWS. So this first project will really help us answer those questions in a very clean system. So it is the development of a mouse model in which we can turn the genes on and off really cleanly and start to ask these questions. So this will be a mouse model that is born with the SNORD116 gene, so those really important genes silent, but with a genetic switch in there so that those genes can turn on later in development or in different areas during development. So Dr. Resnick is going to use this model to address some of the critical questions about, what would happen if you turned it on in an infant or, a baby mouse in this case, or in an adolescent or in an adult. You get a benefit from that. We're excited about this because Dr. Resnick is a very experienced PWS researcher. He kind of knows the genetics and imprinting of PWS as well as anyone, and so he has a really deep understanding of how to make this model work, which is not easy to do. And I think the model is going to be really useful for many in the field to establish the feasibility of genetic therapy and for us to understand the promise of this approach for our kids eventually.